We can, thank you.
Recording.
Great. Well, Josh, why don't you kick it off for us?
Absolutely. Well, I would like to welcome everyone to what is our fifth Learning Health System Tech Forum. This is run by the Learning Health Community, which is we are a 501c3 nonprofit organization. Our very mission, as far as I know, we're the only nonprofit, like our entire mission is about learning health systems. Realizing them according to a set of shared core values that was developed almost 10 years ago, which is amazing to think about, to really try to, one is realize learning health systems at all levels of scale and keep improving them. Second, really, is trying to make sure that they're built in ways that can that they can be brought together to something bigger than the sum of the parts. And again, third being, and maybe most important, although I said that third was this idea of ensuring that what gets done is done according to this core values that make the world a better place. So with that said, the Tech Forum was really very much a brainchild of Dr. AJ Chen, and with a ton of support from Dr. Sophia Wang, who's going to be our fearless leader for this Tech Forum. It's really about bringing together, again, folks that work in technology and entrepreneurship and kind of the innovation space with the folks that love healthcare and learning health systems and health, and bringing them all together to try to learn from each other, and again, also do something even bigger than any of us could do alone. So with that said, I think AJ will welcome our very distinguished speakers from places like Google and Verily and Stanford, and we are grateful to all of you and grateful for the crew that makes it happen. I will, I think I'm now turning it over to AJ, who will make a few comments about learning health systems. Well, after I do that, I'm going to post a couple of links in the chat for people who are just joining to the program for this, to our Tech Forum webpage to learn more. And I will now turn it over to AJ and thank everyone for participating, and thank you all for the work you're doing to advance learning health systems and for coming together to do this.
All right, thanks, Josh. Hi, everyone. Welcome to this forum. My name is AJ Chen. I'm a co-organizer of this forum. And I'm just going to give you a quick introduction of the learning health system vision, and then a series of forums. Basically, learning health system vision was created by the IOM, or National Academy of Medicine, about 10 years ago, and it's designed to transform our healthcare systems to a better care at lower cost. You can see a lot of reports from National Academy of Medicine on their websites. And I just want to point out the three characteristics that I think are most important in my opinion, to the learning health system that we're trying to build. One is the continuous learning and improving of the system, and then the second is really solving the rapid dissemination problem, so that we can actually disseminate the stuff, the best practice quickly. And the third is the more automation at a system level, so that it can be much more cost effective and efficient. So based on this, next please, Sophia.
Okay.
Given this vision, we have been organizing a Learning Health System Tech Forum, and in the past we cover the vision, the infrastructure, interoperability, and clinical research network. And today we'll continue to discuss the clinical research network, because it seems such an important area and topic. Going forward, we were going to continue covering topics at several levels. At the application layer, we talk about the AI tools, research network, dissemination, health equity, etc. And at the knowledge layer, we'll talk about the more technical aspect in terms of machine learning, computable biomedical knowledge, artificial intelligence, PCT, etc. And the data standard is also important at a data layer, and there are tons of data standard out there, and the whole learning health system will be built on all of the international standards. And of course, cloud computing is also important in this in the future of the learning health system. I think that's my quick view of the learning health system and what we're going to do with the forums. And now we going to the main program. Sophia.
Great, so I'll have the pleasure of introducing our speakers. I believe we're starting with Dr. Mahaffey, Professor Mahaffey, who is a professor of cardiovascular medicine at Stanford University, among many other of his leadership positions in our School of Medicine for research. And he has a great interest in multi-center clinical trials, and he is going to speak to us about Verily's baseline health study platform. So join us in welcoming Dr. Mahaffey. I will put his slides up here.
Great. Well, Sophia is doing that. Just thank the organizers for having us today. I'm really looking forward to the discussion. I'm going to spend maybe 15 minutes or so giving you an overview of what the Project Baseline Health Study is about, and maybe focusing primarily on the acquisition of the data, how we're leveraging those data to understand certain biological processes, but also how I think it could be leveraged in the future for both clinical trials and as well as clinical care. Next slide. These are my financial disclosures. The one applicable here is that I do receive research funding that covers a portion of my salary from Verily because of my work on Project Baseline. All of my financial disclosures can be found on my faculty profile at the URL below. Next. So we're all very aware that one of the main challenges we have right now in clinical research and clinical care is actually defining what a healthy state looks like. And next slide. And as we grow older, sometimes it happens at younger states, there is often a transition from health to disease. And much of what we do in biomedical research is develop strategies for treating the disease when it becomes prevalent. And the challenge that we have, and I think the great opportunity that we have, and what Project Baseline is focused on, is can we identify a truly healthy state with all the normal perturbations across society where everybody's a little bit different, and importantly, leverage those data to identify the transitions that occur from a healthy state to a disease state, thereby opening opportunities to maybe develop strategies that are better than our current prevention strategies, and develop mechanisms by which we can detect disease at earlier stages than we can now, so we can intervene in earlier stages of these processes and hopefully improve clinical outcomes. Next. So the fundamental objective or goal of the Project Baseline Health Study, which is a partnership between Verily, as well as friends and colleagues from Duke University and Stanford Medicine, was to develop this coalition of interested investigators, and importantly, as I'll get back to later on, the participants themselves, to develop a gold standard of how we can collect data, as well as the tools and technologies that we can use to analyze that data to create a holistic, aggregated view of health, but also the transitions from health to disease, as I've outlined already. Next slide. Riffing a little bit on Google Maps, Google has done an incredible job mapping the world. We all use it, or similar applications like it, to go to the many places that we visit daily or on vacations less often. And our audacious goal was, can we map human health with that kind of granularity so that we can really achieve our goals of identifying the transitions from health to disease. Next slide. Well, you're sitting there wondering, well, other people are doing this. We have the Million Vets Project. We have the UK Biobank. We have all of us through the NIH and other collaborators. So what is unique in our minds for Project Baseline Health Study? Well, when you're doing these kind of cohort longitudinal follow-up observational studies, you have to ask yourself various questions about what you want to focus on. One is, you can be incredibly broad with a wide breadth of types of patients that you want to follow. You can follow 1 million vets like the VA system is doing right now. Next slide. You also need to decide how deep you want to go in your assessments of each of those patients. So in the Million Vets Project, they're not going very deep. They're leveraging the VA health system and the EHR that's part of that health system, and then doing genetic analyses, collecting blood samples for genetic analyses on these participants. And then finally, you have to ask yourself, next slide, how long a follow-up do you want to do? So something like the Framingham Heart Study, which is now entering its 3rd and 4th generations of follow-up, has gone extensively over time, but the breadth is not that great. It's just the residents of Framingham, Massachusetts, and the depth is modest. And when we designed the Project Baseline Health Study, we wanted it to be the deepest dive that has ever been done in a cohort longitudinal follow-up. And I'll let you decide if we've achieved that goal. We wanted to have modest breadth, and then we wanted to continue to follow patients over time, as you'll see. Next slide. So who did we enroll in this cohort study? First and foremost, through the collaborators at Duke and at Stanford, we wanted to make sure that in those locations, we enrolled a population that was representative of the United States in terms of key demographic characteristics such as age, race, and ethnicity to mimic the US population based on recent census polls. We also wanted to make sure that we enrolled a cohort of healthy people. Obviously, we need healthy people to find the healthy state, as I mentioned earlier, but we also wanted to overpopulate or oversample for people with specific diseases or at risk for those diseases based on traditional risk factor tools and assessment strategies available now. Because cardiovascular disease is still the #1 killer in the United States and around the world, we wanted to have an oversampling of people with or at risk for cardiovascular disease. And because cancer is a leading cause of death as well, we wanted to look at lung cancer and breast and ovarian cancer. Next. And how did we follow these participants? Well, the initial aspect of the enrollment was all through web-based platforms where people could learn about the project. They could read about what our goals were, what their roles would be within the project, and then they could do their initial consenting and screening online. We then put those people that showed interest into a waiting room and then enrolled them over time to come in for their first visit, which was a comprehensive on-site visit that lasted 2 days and 6 to 8 hours of assessment on both days. In fact, we collected nearly 6 terabytes of data on each individual during their initial visit. We then did annual follow-up visits, and in fact, we're just finishing up our 3rd year of annual visits. with a similar assessment, not quite as comprehensive as the initial assessment, about a full day of evaluations. And then between the initial visit and the annual visit, we had continuous monitoring and reporting of information from the participants through several different strategies. One was through web-based or app-based tools where we asked the participants to give us information about their diseases, medical care, hospitalizations, as well as quarterly surveys about mental health and other important conditions that they faced. We also had an investigational watch that the participants wore that we collected data from. And then the circle there is a sleep sensor that was put underneath the mattress to monitor sleep cycles and other important information. So this was an initial assessment in person, annual in-person visits, and then continuous monitoring using both wearable and digital technologies, but also web-based applications. Next slide. Well, this is why we think perhaps we are one of the world's deepest data sets on human health, and it was our strategic approach. These are all the categories of the types of information that we collected on the rows, including medical history, routine clinical labs, omics, proteomics, genomics, metabolomics, obviously vital signs, and then a comprehensive selection of imaging, both cardiac imaging and physiological assessments with stress testing, but chest radiographs, eye examinations, as seen here with both OCT as well as corneal topography and fundus photography. And then we also did a number of physical assessments. And I think very importantly, and as we've seen with the pandemic and the challenges that we face with mental health issues, a very comprehensive set of mental health assessments, both in person as well as online. And then a number of lifestyle data that were collected as well. And also knowing where these people live, we can link in environmental exposures. We, as many of you may know, had really intense fires here in California a year or so ago, and we were able to collect information from environmental exposures as well. Next. So when you think about all this data, we really thought about it in three different domains. One is how we collected all of this 6 terabytes of data on every participant each year. Importantly, how we could organize it. And then, most importantly, how we could activate those data to do important analyses and assessments of health states, transition to disease states, so that eventually we will be able to identify important observations that could lead to improvements in things like prevention, detection of disease, and treatment of disease on down the road. Next. As I mentioned early on, I was going to get back to this concept of the importance of the participants in this research effort. And I would also pose to you, in all of the research efforts that we all undertake, just like we have shared decision-making in clinical care, I think we need to have shared decision-making when we are thinking about designing clinical research projects, conducting them, analyzing them, interpreting them, and reporting them, to keep the participant central to that process, as well as the scientists and staff and other stakeholders and collaborators. So in Project Baseline, we actually did a set of comprehensive advisory groups, surveys, and other assessments of potential participants to help us design an experience, both through the platforms, but also in person, that would be most well received by them. But it just didn't start at the beginning. We have thought through comprehensive strategies to show continued appreciation for the participants, because this project will only be successful if we're able to keep them engaged longitudinally to continue to collect the information over time. And then we wanted to find ways to show their value to the program. And one of the really interesting things that the participants want, and we know this from other surveys as well, is they want to see their data. And so that we have a whole group that is thinking about how we return the results to the participants in a way that is well received, they understand it. But as you can imagine, returning a lot of medical information to participants also has an incredible amount of responsibility with it, so that it's done in a way that's appropriate, understandable, and that participants, especially when they have abnormal findings, have appropriate follow-up outside of the research team, because we're conducting the research, but they have to have a connection to the clinical care. Next. And so I think of a simple mnemonic to remember this, and that's the 3 E's. You've got to engage, you've got to educate, and you've got to empower the participants in research projects like this and others to get their dedication and commitment to the project to enhance the data that you're collecting and the scientific rigor of what you're trying to do. Next slide. Well, as you can imagine, with the 6 terabytes of data that we're collecting on these participants at the beginning, and then multiple terabytes of data on annual visits and during the routine monitoring, we have an enormous amount of information that's being collected in terms of biospecimens, number of hours of sleep data that we have. We did grit testing, we did stand and walk testing, we collected symptomatology, we did 6-minute walk test and 10-meter walk test, where we have millions of meters of data collected. Next slide. So what are we doing with all this data? Well, and strategically, we waited a couple of years because we did want to have some longitudinal follow-up of the initial cohort, as you'll see in a minute, of 2,500 participants. We formed a governance around this data, and we decided that we were going to focus on certain key areas to start with. Obviously, the cardiobolic working group, because that was one of the areas that we wanted to focus on with the oversampling of people with cardiovascular disease or at risk for cardiovascular disease. We really moved mental health into the forefront, particularly with the emphasis that has been placed on mental health, particularly through the pandemic. We have an entire group that's looking at return of results, as I've already mentioned, thinking about strategies to do that in a responsible way. And then we thought about a unique approach of what we were talking about, rewriting the textbook of medicine. You know, we have so much data on these physiological assessments that we all do every day in clinic. We look at resting heart rate, we look at resting blood pressure. What does that really mean? And so now we have this data along with all of the other information that we can start looking at important correlations and associations of resting heart rate with other data that we're collecting. I think Brad's going to talk a lot about this in his next, in his talk, the electronic health record working group, because we're collecting a lot of this data, if not all of this data, outside of the EHR, but we're looking at ways that we can start leveraging the EHR data that's being collected on the Project Baseline Health Study to see if we can maybe develop a more virtualized component to the follow-up of these 2,500 participants. And then coming soon is the oncology working group. And we're really excited that over the last year, when we devoted the effort to these scientific inquiries, that we're starting to publish papers in the scientific literature, but we're also thinking of ways that we can share this information and disseminate these findings and observations in other platforms so that we can get the information out to broader groups of people than those who read the scientific literature. So stay tuned. And forums like this we're really pleased when we can have an opportunity to share with you our initial efforts. Next slide. Next. So this is the initial 2,502 participants that were enrolled. They were enrolled at 4 sites in North Carolina and California, Palo Alto at Stanford, Durham at Duke University, a community in central North Carolina called Kannapolis that's managed by Duke University, and then at the Chile Foundation in Southern California. And you can see here the breakdown of age, sex, and race and ethnicity in the initial 2,500 participants. A quarter of the people were over 65 years of age. We thought that was important, especially as we're looking for transitions to disease. More women than men, which is really important. At least in my field of cardiovascular research, women are notoriously underrepresented in clinical trials. And then we were able to actually hit our targets in terms of the race and ethnicity breakdown, which really mimics the entire US census. Not necessarily what you would see in Durham and the Bay Area, but really what you would see across the United States. Next. I'm going to share with you a couple of analyses that we've done so far to give you a sense of what we're trying to do with the initial analyses out of this data set by the working groups that I showed you already. The first deep dive was looking at a study of the PHQ-9, which is a survey tool for depression that's used routinely in clinical practice. And we really wanted to start looking to see what some of the associations were of PHQ-9 with different clinical characteristics and demographic information, and then do a series of analyses using a variety of statistical methods to look for more detailed associations with the other factors and data that we've been collecting with PHQ-9 scores. Next slide. And here you can see the analyses for some of the learnings from the PHQ-9 investigation. You can see a comprehensive listing of many of the factors that we looked at and the association. And you can look at the broadness of the bars is the strength of the association. And the summary is that younger participants in the study were more likely to have higher PHQ-9 scores and therefore higher levels of depression. Not surprisingly, lower socioeconomic status, including those factors that you can see here, were associated with worsening depression. Smoking status and several of the patient-reported outcomes and conditions that you can see on the right-hand side of the slide. Next slide. We're now taking the way that we did this for PHQ-9 and attacking a number of other important issues that I think we need to understand better, looking at cardiometabolic candidates. As you can see, I've already mentioned resting heart rate, 6-minute walk test, coronary calcium score that's being used more and more now in clinical practice. We're doing analyses with GAD-7, which is an anxiety assessment score scale that is often used in clinical practice. And then a number of physical function parameters that we want to see how function in the participants correlates with other important biochemical, biological, socioeconomic factors. Next slide. And here's just an example of the resting heart rate. Our initial look showed that participants who are on average more active by 1 hour a day had a lower average resting heart rate of about 2 beats per minute. People who slept more had better lower resting heart rates. And now what we're starting to do is additional analyses like we did with the PHQ-9 to really understand the associations between a variety of other clinical and demographic factors and the resting heart rate. Next slide. A really interesting set of analyses being run by my good friend and colleague out of Duke, Svati Shah, is looking at predicting how old you are based on your DNA. And there's a concept of DNA methylation analyses that actually shows how your genes are being turned off and on at various times. And the amount of DNA methylation gives you an assessment of what your current age is. And then it can be compared to your actual chronological age. And what's been shown by others, and now we've demonstrated it in the Project Baseline Health Study, is that there are a group of patients or participants that are much older based on their DNA methylation than their chronological age. And there's also a group of participants that fall below the blue line that are actually younger by their DNA methylation than their chronological age. We want to understand those groups of participants better. We also found in follow-up that there were a group of patients that aged much more during one calendar year. They may have aged 4 or 5 years based on their DNA methylation. And it would be really interesting to go back and see what was happening with that group of participants. Next slide. And then finally, we're trying to engage directly with the participants to help with some of these scientific inquiries. Last year, during the spring forward, when we set the clocks forward, we asked people to be much more diligent in monitoring their sleep and asking a series of questions on an app-based platform. Next slide. And we really found that around the time of the spring forward, or setting the clock forward, that the amount of sleep that people got decreased, as you can see here. We also found that they had less satisfying sleep and felt less rested. So now we need to understand what these participants were doing that didn't feel this, that may be different than the participants who did not feel as rested during their time frame, and understand if there's potentially interventions that we can do to decrease sleep interruptions or disruptions during the times when we set the clocks forward or back. Next slide. Well, I've been showing you a lot of data, and we're still not quite sure exactly what we're doing at times, but as we continue to explore these data, both with investigators from Stanford and Duke and Verily, but also including others, we're really excited about the potential for the future. Next slide. And I think this is really the time where we have an incredible opportunity. We have an unprecedented amount of data here, but we also now have the tools, both the IT platforms, but also the statistical methodologies that have been advanced over the last several years to really think about how we can organize these data, but also analyze it in new and exciting ways to activate on it in the future. So I will end there with just the thought that we believe that the Project Baseline Health Study, that it is a continuous follow-up, a longitudinal assessment of people, a deeper dive in understanding biology than any other group has done to this point in time, and a very connected community of researchers and participants that's going to allow us to do some exciting things in the future. So thank you very much, and I'll hand it back to you, Sophia. Thank you again.
Thank you so much, Dr. Mahaffey. That was great, great overview. A couple questions came up in the chat, and I'll just ask one and hold the others for the end. One question was, did all participants have to participate in all components of the data collection, or could they opt out of certain aspects of continuous monitoring or DNA or something like that?
So the participants were able to opt out. We asked specifically about whether or not they wanted us to collect their DNA. Some participants may not want to do certain things. One of the most controversial was actually stool collection. And many participants didn't feel comfortable giving stool samples. So yes, we tried to collect the data comprehensively across the entire population, but participants could opt out.
Interesting. All right. Great. So we'll hold the other questions for the end. In the meantime, I will introduce our second speaker and make sure that he has a chance to get through his content as well. We're very fortunate to have Dr. Brad Hirsch with us. He is a CEO, co-founder of Signal Path, which is now part of Verily. So he has a great passion for conducting trials and trial platforms and designing all of that. So we are very, very excited to hear about his vision for how these clinical trial platforms can help with the learning health system. So with that, I will stop our share and have Dr. Hirsch share his slides.
I appreciate it. Can y'all see my slides and hear me?
Yep.
Perfect. So always difficult to follow Dr. Mahaffey. I am relatively new to Verily, but I'll give you a little bit of view of how the baseline health study was incredibly deep in the work that's been done. And obviously that can't be replicated across the entirety of the country. The complexity of what it took to pull off that work that he and others have been doing for years now is really the underpinning of the broader evidence generation infrastructure that we're looking to build at Verily moving forward. And I'll talk about what that means and how we're leveraging it over the next 10 or 15 minutes. And I'll do my best to save a couple of minutes for questions at the end. You know, this all gets to the idea that in clinical evidence generation today, we have the traditional phase 1, phase 2, phase 3 infrastructure followed by approval. And then post-approval of these agents, there is a void. There is some phase 4 data, but it's not adequate to truly understand how do we utilize these agents in the day-to-day execution of practice. You know, often the phase 1 through phase 3 trials that we do are in very specific patient populations, very controlled. And then, you know, in my world of oncology as a medical oncologist, so many things are used off-label and with inadequate information. And so part of what we're doing, the baseline health study, was really a training to look at how do we best bridge that void, bridge the inadequate information that we have out there to guide care and optimize things in the setting of a learning health environment, a learning health system that we've been talking about for years. And if anything, the approval mechanism for many of these drugs from a regulatory perspective are moving approvals earlier. And so we'll only have less information over time instead of more, particularly in areas like oncology. The positive is that there are big regulatory and other changes that are happening. The Technology Modernization Act, 21st Century Cures, including 2.0, STAR, and other programs that are really laying the infrastructure to generate the type of data we're talking about at the baseline health study, but looking at novel ways to do it and really optimizing that. And in particularly in the last couple of months, it's been pretty fascinating to see the amount of guidance that's come out from the FDA, one of which is shown here. The real world data guidance around assessing EHRs and medical claims data to support regulatory decision making. And so we're really in an interesting moment when we're starting to take a step back and say, you know, not everything we can do can be with the death of the baseline health study. How can we leverage our learnings, leverage infrastructure like that to really reimagine what it means to go through that regulatory decision process? Similarly, MHRA has come out with recent guidance. We were part of a series of National Academy workshops last year around what the clinical trial enterprise of the future should look like between January and May of last year, to again try to say, what does evidence generation of the future look like to meet this? You heard Dr. Mahaffey just speak at length of this idea that at its core, we need longitudinal health data. We need to, you know, we come from a clinical trialist perspective, and the baseline health study is a really interesting change in that view of not just a very specific question where we go incredibly deep with incredible structure, but how do we actually look at that longitudinal journey of a patient, which they've done with incredible skill across the 2,500 patients within the baseline health study. But when we look at the different ways that we can leverage the baseline health study to start comparing other ways to aggregate data and really understand what needs to be in the infrastructure of a trial, what needs to be in the infrastructure of the baseline health study, the learnings we're seeing, but how else can we augment that as a training model has been a really interesting one. So there's the clinical trial model that we've talked about and we all know well. There's the EHR data at health systems today that's often episodic and changes based on where your care is being delivered and how you engage with the different systems. There's claims data that exists today at scale. And then there's biologic data, whether that's genomic data in oncology and elsewhere, that are sort of the traditional ways that we first started with of thinking about how do you replicate things like the baseline health study and think about that at scale. I was at Flatiron Health with Amy Abernathy and others in the past. It seems like so long ago when we were first talking about what does research grade clinical data really look like and mean, particularly in the setting of an EHR. I would say there are now innumerable companies out there that are doing that well and doing that differently. And now it really is, how do you aggregate that data and how do you understand it? Because it's at its core, as we look at augmenting things that we don't do with the depth of the baseline health study, you have to think about traceability, completeness, accuracy, plausibility. And often you're bringing together different data sets that have very different ideas when it comes to those elements. In an EHR instance, you have to understand what is the completeness of that data for a given element that you're looking at the individual element level. How do you represent that so folks understand the data? How do you have regulatory agents see that? And how do you train that against gold standards that you have, like the baseline health study? And so that's a lot of our work initially at Verily right now is around aggregation across data sets and training against gold standards like that. And in that process, one of the things that we've focused on fundamentally is this idea of permission. How do you consent people and engage patients in the way that Dr. Mahaffey and others did very well on the baseline health study, so that they are participant in that journey, so that they're agreeing to give you the data that you have and helping you to organize it. So you have to understand the periodicity of that data, the completeness of that data, and organize it in the right way, acknowledging that there are gaps. There are times when people might not give you permission to see it, and really organize it and then document the underlying data quality, provenance, and other elements, improve it, and then really leverage that to use it based on that longitudinal work that we've all been a part of. At Verily, and I know I'm going quickly, just I'll give you a broad overview and then I'm happy to dive more deeply into any of this. But the way that we're thinking about it is, first, we need the trial software platform. We need the kind of work that the team has done related to the baseline health study. Because at the end of the day, you need to get permission from patients through e-consent enrollment. You need to be able to understand their experience, e-pros and other elements, surveys and other elements. Where relevant, you need to be able to get CRFs, virtual visits, remote patient monitoring. You need that core infrastructure for these longitudinal registries that everybody wants to see, that'll really allow you to learn in real time and have that LHS infrastructure. I have a bias. We also need to engage health systems, like many of y'all, very deeply. So I came from, prior to joining Verily in last September, I led a site CTMS company called SignalPath, where we spent the last decade really putting infrastructure across health systems that coordinators, regulatory, finance, legal, and all use across their portfolio of trials to operationalize those trials. And we became the tool that a coordinator uses with the patient and the doc, that finance uses to build their budgets, et cetera. And that became very powerful for us, because when you talk about the setting of a learning health system, being able to truly have research grade infrastructure at health systems to test different care pathways at different agents and have a scalable infrastructure to do that becomes incredibly powerful. And that's what led SignalPath to join Verily. So if you have the data infrastructure to be able to collect intentional data, consent patients, et cetera, you have the health system infrastructure to do it. And then you look at different ways to bring together data sets that you can get from any different place, EHR, claims, biology, et cetera. You become a really interesting way to replicate aspects of what we've done incredibly deeply in traditional trials and baseline health study and others, to then really have what we believe is the evidence generation infrastructure of the future. And that gets to this idea that built on our trial platform at Verily is now this longitudinal data layer that is aggregating data that we intentionally collect, that we intentionally engage health systems to do, but also that's coming from these other areas. And just give you a couple of ideas of examples of that at Verily. We have a study watch that's on its 3rd generation that not only captures the traditional sensors you would expect to see in a research grade watch, but also can guide a patient through a 6-minute walk test and the different aspects, so that we can have a very structured way of getting that data. We can generate e-pros on the wrist and different ways to engage folks to really get longitudinal research grade data. And it's something that we've spent a lot of time thinking about. The ultimate goal, though, gets back to the baseline health study, which is then, how do you tie all this together into a study design layer where you can really reimagine what it means to do a study and move to a different iteration of that, where, in our view of the world, it's not a decentralized clinical trial versus an on-site clinical trial. It really becomes a fit for purpose element of asking, there's a cardiac device that needs to be implanted. There's an oncologic agent that needs to be delivered that has to happen in the clinic. There's a certain patient population that needs to be recruited in the clinic. There are other elements that could happen at your local CVS or in your home. There's another element that can happen on your study watch and on your phone. So we're really having the right conversation about where should a given activity occur, what is the right way to generate data, so that we can reimagine, we can use the work we've done historically as the gold standard, and then reimagine what it means to do that work. And Shannon Sullivan, who's on the phone, has spent a lot of time on the Verily side thinking through how do we leverage a lot of what we've done historically based on health study and others to start thinking of what are the most important learnings that we have, and how can we recreate that using other elements that are out there, and get away from this idea of, again, a decentralized trial versus a site-based trial, and really think about what is the evidence generation of the future look like? What is fit for purpose for a given regulatory intention versus what's fit for purpose in the learning health system of care pathways and others that we're just optimizing, and what is the data quality that you need to do that work? What is the data integrity you need to do that work, and make it much more intentional of how we think about it. So in the end, this is the goal. The goal is to say, we're going to move approvals earlier. We're going to have phase 1 through phase 3, but then we're going to leverage our experience. We're going to leverage the data that exists or that we can generate across a myriad of different projects internally to Verily, tools that we and others have developed, data sets that exist internally, to really drive things forward and help ultimately iterate on care and drive the health of the individual forward. I intentionally went through that relatively quickly just to try to tie together what we've been talking about today, and hopefully in the next 10 minutes, we can have a conversation about this and where we're going with it. I appreciate the opportunity to present today.
Thank you. Thank you so much, Dr. Hirsch, for giving us that great overview. And I want to circle back now to some of the questions that had come up earlier in the chat and kind of riff on them a little bit. And one of the earlier questions was related to baseline, project-based baseline, but actually gets at a larger question. So the original question was, how do you oversample from underrepresented populations and earn the trust from these groups, have them give up such deep data about themselves when they might be distrustful of research due to historical transgressions? And I think even beyond that, even if you're not an underrepresented person, member of a population, how do you kind of assuage people about the privacy of their data, whether it's going to be used for commercial purposes, who has access, especially when kind of Verily or Google, a big name like that, is involved.
Yeah, maybe I'll take it from the academic side, and then I know some of my collaborators from Verily are on the line, and if they wanted to comment as well, I think there's two streams of thought here that are part of that question. One is just how you logistically oversampled for certain groups. And that was relatively easy for us in Project Baseline because, as I described, people would go into the portal and express interest in this. They would consent online and provide us with some information. And then they would get put into a waiting room. And then we were able to invite them to come for their initial visit. And we could select people of certain ages, certain race, certain ethnicity, and other factors. So we were able to sample in a way that we could achieve our goals of a broad represented population. Now, the second one is obviously really challenging. And there's a lot of research that have gone into how you gain trust in clinical research participants in general. And we did it in a multifactorial approach. As I mentioned early on, we got a number of participants to help us design the protocol, design the consent form, design the platform, so that the patient or participant facing data and information had all been vetted by potential participants so that they understood what was being asked, and information being provided in a way that was meaningful to them. We also had a number of outreaches to clinicians in the area where we were recruiting. And so that participants could be approached by their own clinical providers or given information about Project Baseline from their clinicians, because we know from other data that patients really trust their doctors. And if their doctors tell them that a research study is a good thing to do, they're much more likely to do it. And we also had community advisory boards that we presented the information to. And community advisory boards, both of people in the population, but also of patients with certain diseases that we would want advocacy to help us understand how to approach those particular patient populations. So a very comprehensive approach. I don't know, Shannon, I see you're on, or Brad, if you wanted to comment, or others, please do.
Yeah, Ken, you described it beautifully. I just wanted to add, so I'm Shannon Sullivan. I'm a lead clinician for baseline health at Verily Life Sciences. And, you know, one of the really special things about the baseline health study effort is the two-way flow of information and learnings as we go along. And so there is you know, these participants not only have in-person relationships with their local site, but there are also opportunities for them to gather together to learn about some of the research that's coming out of baseline health study. There are annual retention events or quarterly newsletters. So, and on the participant side, there are also opportunities to share their thoughts, their ideas, their feedback about the tools that we're using on an ongoing basis. And so we've really tried to be very intentional about including the participants and the participant voice in really the design of how we move forward, and to be able to keep that participant front and center.
And the only thing I would add is, you know, on the signal path side, one of the things we've had a lot of success with is we have a lot of faith-based organizations and others that have been a part that when we intentionally select the right sites, the right health systems that have deep experience in that space, based on where they are geographically and other elements, we've had a lot of success at being able to engage the right populations and make sure that there's broad representation on the work that we do. And it's something we're looking forward to only going deeper with prospectively here at Verily.
Great, great. Another question that I had was, you know, Dr. Hirsch, you alluded to the messiness of the data being collected, you know, in sort of real world settings. And of course, traditionally, we think of clinical trial data as being incredibly clean and curated, often due to intensive human labor. So, you know, in your platforms and the tools that you've built, are there special tools to help clean that messy real world data at the point of collection? And maybe can you talk a little bit more about how you handle that and what makes that special?
Yeah, and I'd go back all the way to our days at Flatiron. There were a couple of papers that Amy Abernathy and others, and I and others published around how do you even think about research rate data? What are the 10 attributes of research quality data? And really understanding what does that mean and how do we represent it? Because I think as much as anything, the data is all, there's no way to fully address the messiness of EHR and other data, but it is transparency in what it looks like, agreement on the standards that are fit for purpose, and then allowing people to do that. I had a great conversation. One of the positives of the Google world is we had an interesting conversation with a scientist at Google yesterday whose whole job is data visualization. And I think one of the biggest gaps is helping people to understand and engage with data quality, data integrity, and other elements. And so he was showing us the 50 different ways that you can show a patient's journey and understand where there's missingness of where they moved from New York to Florida for the summer, and how to visualize those certain elements so that people can engage. Because I think one of the biggest difficulties that folks have is understanding, you know, if you don't have a deep background of doing that work, how do you understand and engage and understand the fit for purpose for a given data set based on sort of agreed upon rituals and approaches? And so one of the things that I'm most excited about that I was hearing about recently is really diving into helping people understand that and engage with it.
Yeah, and Sophia, if I may, the one thing that Brad and I have talked about for a long time, right, is being a clinical trialist. The beauty of we can be successful in collecting the data through the EHR in a way that is of high enough rigor and integrity to really start asking questions in a randomized strategy. That would be the ultimate goal. And, you know, recently, for example, the adaptable trial, which was funded by PCORI, that enrolled 15,000 people to asking the question of which dose of aspirin is right. And they did it all through EHR and the PCOR net common data model that was set up across a number of different institutions. So we're showing that this process can work. It takes a huge investment, but eventually I think we're going to get there. It's just going to take some time.
Yeah, and extension of that comment, one other thing I would say is not every element of every clinical data needs to be of the same quality and of the same depth and rigor. And so one of the areas when I was talking about novel ways to generate data, whether that's through a biomarker or a phone or whatever it is, if you can augment certain aspects of the data set to make sure the rigor exists there to compensate and make sure that, you know, there are other elements that you don't, and being very intentional about that and understanding that is another way that we can sort of in real time augment what is otherwise passively collected to meet some of the expectations that Ken was referencing.
And I imagine that, you know, not every health system is going to have a sort of an EHR or data infrastructure to support, you know, pulling out all of the data that we want. And one of the audience questions is, you know, what are some remaining big rocks that you need to move to get more health systems to buy into the prospect of having kind of a research grade data infrastructure? How do we get to that kind of huge scale?
I'll give one answer, and then I'll be curious as Ken's as well. You know, I think one answer is questioning whether you actually have to go health system by health system to get that data, whether there are other relationships and infrastructures to actually get at some of the data. And we can talk in the last minute. There's probably not time to get too deep into that, but there are a lot of other ways these days to partner and get data, especially if you have consent from patients and they're willing to share through different mechanisms. And so I don't think it necessarily is relying on every health system signing on and everyone having a common data model. That world is evolving pretty quickly. I don't know if you agree with that, Ken.
I agree completely. I mean, I think there's, you know, we got to get back to the fundamental construct that patients' health information is their information. They own it, and they should be able to do with it what they want. And I think Brad's spot on. You know, the other challenge, though, that until we get there is that, you know, we're not like a country like Sweden where everybody has a unique identifier and you can track people across all their healthcare systems. And so right now, because of the mobile nature of our society, we get our care in a variety of different systems, and tracking that data to be comprehensive in your assessments when you're doing longitudinal follow-up is still challenging. So we've got to fix one or the other of those or both.
Great, great. Well, I'm sure we could have a very long discussion on all the different nuances of this topic, but I want to be respectful of our speakers' time. So thank you so much, Dr. Hirsch, Dr. Mahaffey, for giving your excellent presentations and teaching us about Project Baseline and Verily's clinical research platform. And thank you all to the participants as well who signed on and for your great questions and participation. And we'll hope to see you at our next forum.
Bye, folks.
Thank you. Take care. Be safe.
Thank you.
Everyone.